Good afternoon. Good afternoon, everybody. And welcome to the MHRC seminar. It's my pleasure to introduce Dr. John Thyfault this afternoon to our seminar series. I've known John for a while, and it's great to have him join us this afternoon. John did his undergraduate work at Fort Hays State University in Kansas, Western Kansas. And he also did master's and a Bachelor of Science there, and then did his PhD at the University of Kansas in Kansas City. Then went on to do a postdoc at East Carolina University. He worked with Linus Dome, who we know well as a pioneer exercise physiologist. He then got hired at the University of Missouri in Columbia, Missouri, where he spent 10 years as an assistant and associate professor. And then he was recruited where he is now, to the University of Kansas, his own alma mater, where he did his PhD. And he's now been there for 5 years. And I looked through John's biosketch. He's extremely well funded by NIH, and he's always been interested in metabolism. And what excited me in particular was his work on liver, which I thought was a novel field to look at during exercise, a novel area to look at during exercise. And so really excited to have John join us. So John, if you don't mind sharing your screen with us and turning on your video, and I'd love to hear you talk about aerobic capacity, skeletal muscle mitochondria, and risk for disease. John, welcome.
All right, thank you. Can you hear me?
Yep.
Okay. Thanks for having me, Dr. Hood and everyone at York. I appreciate this opportunity. Trying to get my slide to advance here. There we go. Okay, so my outline today, I'm going to show 3 different lines of data, or 3 different lines of research, really focused on the links between aerobic capacity and skeletal muscle health, mitochondrial function and skeletal muscle. And I'll throw some liver in there as well. So we're going to talk about a little bit of early data we have on Alzheimer's and skeletal muscle, some work we're doing on exercise and pharmacotherapy, specifically statins, and then our work on liver metabolism and how skeletal muscle mitochondrial function and metabolism is really important to be synergized with liver metabolism. And all of these things, of course, affect aerobic capacity, which is the best predictor of health. And so here you go. First slide. This is classic data now from Steve Blair, showing that aerobic capacity is a great prognosticator of early mortality. And so in this study, what they showed was that irregardless of any other factor, if you were in the low aerobic capacity group, you had the greatest risk for early mortality. And if you were in the highest aerobic capacity group, you had the best protection. And then importantly, even if you're just moderate, you can see there's a huge benefit to just having moderate aerobic capacity. And several groups have reproduced this data in different cohorts of people of all socioeconomic statuses and races. And importantly, they've even gone on to show that this is something that's modifiable. So this is a study where they looked at 5 years in between measuring aerobic capacity. And they had people in that 5-year period who were unfit at the beginning and stayed unfit. They had people that went from unfit to fit, and they had people that stayed fit. And you can see, even if you were able to increase your fitness over that 5-year period, that your mortality risk went down. So this is a really strong area of research. There's some epidemiologists who say that, or the data would suggest that this is the best prognosticator of early mortality is fitness. And so if we talk about fitness a little bit more, aerobic capacity, if you're not familiar, I think most people on here are familiar, but this is what we talk about with VO2 max. So it's the maximal volume of oxygen that someone can consume during max intensity exercise, which of course we can do on a graded exercise test to failure, either on a treadmill or a bike. And of course, the amount of oxygen that you can get into the system and use during maximal exercise is dependent on the pulmonary system, the cardiovascular system, and then skeletal muscle, where you actually have the muscles contracting and consuming the oxygen to fuel the exercise. And it's also important on circulating factors like plasma volume and red blood cell volume, stroke volume in the heart, and of course, oxidative capacity. And VO2 max or aerobic capacity is strongly influenced by your behavior, your genes, and your age. So this is a study where they look cross-sectionally at aerobic capacity in individuals who were either aerobically trained their whole life or sedentary their whole life. And you can see that at a young age, aerobic capacity is much higher. And then if you're active or an active exerciser, it's much higher at a young age. So this would be like bone density at a young age. You want to get as high as you can at a young age. And then you can appreciate that as individuals age, aerobic capacity tends to decline. And even if you're an exerciser, it continues to decline. Now here you'll see, if we pick an arbitrary point across time here or a level, that there's about a 3-decade difference at the point that the sedentary individuals meet this maybe at 28 mils per kg per minute versus the exercise trained. So again, if you're able to get your VO2 max up as high as you can at a young age, that's going to confer benefit over your lifetime. Now, there's also a genetic or intrinsic component to aerobic capacity, and most of this work comes from Claude Bouchard's work, including the heritage family studies. So if you look at individuals who are sedentary, they do not exercise, you'll be surprised to see that there is a lot of intrinsic variability in the fitness values of those individuals. So in the heritage study, they recruited sedentary men that were between 17 and 35. Their average VO2 max was 41, and about 7% of those individuals were below 29 and about 7% above 53. So they're really a bell-shaped curve, a lot of heterogeneity. So you can see even in a sedentary condition, there's quite a bit of variability. And then there's also big variability in response to exercise. We know in general that exercise will increase VO2 max or aerobic capacity, especially if it's intense enough. But there's big heritability heterogeneity in how much people respond. So in the heritage family study, they saw that the average increase to a 6-month exercise intervention, 5 days a week, was about 16%. But about 20% of individuals were high responders and 20% were low responders. In fact, quite a few people didn't respond at all. So, and further studies have shown that the genes that seem to predict this intrinsic aerobic capacity are really separate from the genes that predict exercise responsiveness. So there's a lot unknown here, but what I want to highlight is that there's a lot of genetic variability. And overall, it's thought that about 50 to 60% of your aerobic capacity is due to genetics. And I talked about mortality already, but I also want to highlight that multiple chronic disease states have been individually and independently linked to aerobic capacity, such that if you have low aerobic capacity, your risk for these disease conditions goes up significantly. And 3 of those diseases I'm going to talk about today are Alzheimer's disease, the metabolic syndrome, and fatty liver. Other diseases are listed below, including cancer, breast cancer, and colon cancer in particular. So I really want to highlight that one of the salient findings that has been shown by different outcome measures across different labs, across different decades and different cohorts, is that there always seems to be a correlation between the amount or function of mitochondria and skeletal muscle and peak VO2 or VO2 max. And so here's a picture of a pioneer, Bing Saltine from Copenhagen. He led some of these studies. Other groups at Ball State and other places have some data here, but you can appreciate that in trained and untrained individuals, young or old, lean or obese, and even very old individuals, aged individuals, that there's a nice correlation between various measures of VO2 max and mitochondrial function and muscle, or mitochondrial content and function. So Dr. Steve Britton was a cardiovascular physiologist, I think, at the time. I believe that's what he used to focus on. And he was very taken aback by the epidemiological data that aerobic capacity was so important for health. So starting in the late 90s, he decided that he would create an animal model, working with Lauren Cook, that he would create an animal model to better investigate differences in aerobic capacity. And so what he did was he took rats, a heterogeneous population of rats, and he ran them to exhaustion on treadmills with graded exercise tests. And he took the rats that were the best runners, the top 10%, and bred them together. And he took the worst 10% of the runners and bred them together. And he called the best runners high capacity runners, or HCRs, and the worst runners low capacity runners. And they didn't just do this with one generation, they did it over multiple generations. And so in 2005, they published this paper showing that at generation 11, there was about a 350% difference in distance to exhaustion or endurance running capacity between the HCR and the LCRs. Now they're out to about 40 generations, and the HCR rats can run about 2 kilometers before reaching fatigue, and the LCR rats can only run about 200 meters. Importantly, 2 things are important here. One, these animals do not have exposure to exercise. They are only bred for exercise endurance ability. They're not provided, or at least in our studies, we don't provide them wheels or treadmills. So this is inherent aerobic capacity. And 2, they have shown repeatedly that there is differences in VO2 max measured directly between these 2 strains of rats, the HCR and the LCR, and it's quite significant. And what I find to be most striking as we use this model more and more over the years is that they saw the epidemiology that aerobic capacity was important for longevity and lower mortality. They created an animal model to study that, and then the animal model mimics the epidemiology. So here they did aging studies both in Norway and in Michigan, and what they found was that the LCR rats lived about 6 months shorter than the HCR rats, which I just think is profound. So in today's talk, I'm gonna go back to the HCR, LCR rats later. So in today's talk, I first want to move into Alzheimer's disease. I have just a few slides on Alzheimer's. I'm lucky to work at KU Medical Center, where we have an NIH-funded Alzheimer's disease center, and there's 26 of those centers in the United States. We are one that is primarily focused on lifestyle, exercise, and diet, and also on mitochondrial energetics as a cause for Alzheimer's. So the co-director of the center is Dr. Russell Swerdlow, and Dr. Swerdlow has been writing reviews and collecting data for quite some time, suggesting that Alzheimer's disease is not so much driven by amyloid deposition, rather amyloid deposition is just along for the ride, and that the mitochondrial or the disease phenotype is really due to deficits in mitochondrial function and energetics. And some of the best data to really suggest that is it's very well known that Alzheimer's brains have hypometabolism, a reduced ability to use glucose when they're measured in a PET scanner, and that this hypometabolism actually occurs prior to loss of cognition. And so this is one of the reasons Dr. Swerdlow believes that energetics plays a role, and there's a lot of other evidence. Then also Dr. Jill Morris, who's faculty here and was a postdoctoral fellow under Dr. Swerdlow and Dr. Burns here at KU Med, has also done work showing that people with mild cognitive impairment or Alzheimer's disease, they have lower insulin sensitivity measured by clamp, and they have lower aerobic capacity. So that led us to believe that they must have abnormalities in skeletal muscle mitochondrial function that tracks with these, because typically insulin sensitivity and aerobic capacity track with muscle mitochondrial function. And I just want to highlight that Dr. Swerdlow has shown in other non-brain tissues from Alzheimer's patients that they have mitochondrial deficits. They've done this in platelets, and they've also used cybrid cell lines and used mitochondrial DNA from Alzheimer's patients in those cybrid cell lines, and then those cell lines have abnormalities in mitochondrial function. So this wasn't just that harebrained idea that there'd be a muscle phenotype. Now I do want to highlight that again, VO2 max rears its head here as being important. There's been lots of exercise interventions in people with early Alzheimer's disease. A lot of the results are mixed, some showing no benefit, but in the studies that have been done here at KU Med, what they've clearly shown is that in the individuals who do have an increase in VO2 max with the exercise intervention, they have a correlation with that, correlating well with memory. Also, they have data showing in the individuals who actually do get an increase in VO2 max, they have a correlation with hippocampal volume. So they know that aerobic capacity in response to exercise is important for brain health. So what we did was we wrote an R21, and we simply wanted to phenotype skeletal muscle mitochondria from patients with early Alzheimer's, what we'll call MCI, mild cognitive impairment, versus age and weight matched healthy controls. And I have Chris Perry's picture on here because Chris was kind enough to visit Kansas City twice to teach us how to do the permeabilized muscle fibers and the O2K work to do this. And the whole time he was here, Chris talked about mitochondria. I think the only time he didn't talk about mitochondria was when we fed him Kansas City barbecue. So I had to get that in on Chris. And then Dr. Colin McCone, who was a postdoc and is now a research assistant professor in my lab, conducted these studies. And the idea was, again, just a cross-sectional analysis, MCIs versus healthy controls. We had a balanced design with ApoE4 carriers versus non-carriers, and that didn't really pan out to show much, but ApoE4 carriers are people who have a very high genetic risk for Alzheimer's. We did muscle biopsies and looked at respiration. We also measured physical activity via accelerometers because we wanted to make sure that any deficits that we may have seen wasn't due to physical inactivity in one group versus the other. And then we accounted for what drugs these individuals were on. So a lot of people with early MCI get put on a drug called donepezil. There's other brand names, but they are cholinesterase inhibitors, and I'll get into the importance of that in a second. But we wondered, as I said earlier, if because people with early AD have reduced insulin sensitivity and aerobic capacity, if muscle mitochondria was playing a role. So here's the data. I guess I forgot to put the in size on here, but we have I think we have about 15 in the cognitively healthy, 15 in the MCI, and 12 in the MCI plus meds. And what you can appreciate is that the MCI, or the early Alzheimer's patients, do have suppressed mitochondrial respiration. State 3 ADP stimulated is lower if they were not on a drug. However, the individuals that were on donepezil had normalized, if not even higher, respiration compared to the cognitively healthy. And here you can look at the respiration in the different conditions. State 3 was succinate and uncoupled. And you can see there's differences across the board. And I want to highlight that these were mostly individuals getting very good medical care, highly educated, mostly upper socioeconomic status, who knew that they needed to be eating a healthy diet and they needed to be very physically active to offset their cognitive decline. And we were surprised to find that all the groups, the individuals in all the groups got about 10,000 steps a day. So we don't believe this is due to physical inactivity. Okay, so back to this donepezil. It's an acetylcholinesterase inhibitor, and we have questions about how it would affect muscle mitochondria. And I'm looking forward to the muscle physiologists who are listening to this at the end of my talk to give me some feedback on this. But you are all familiar how a muscle contracts. You have a neuromuscular junction. You have acetylcholine that was released into the cleft. It binds to receptors and it starts the action potential down the cell, and that allows muscles to contract. You also have acetylcholine receptors that it binds to, but then you have cholinesterase enzyme in the synaptic cleft on the membrane that when the muscle contraction needs to cease, it is the enzyme that breaks down the acetylcholine and stops the tonic muscle contraction. So this is a drug. These drugs are given to people with early Alzheimer's to potentiate neuronal signaling in the brain. And there really hasn't been a whole bunch done on the effects in skeletal muscle or in other tissues that I can find from a basic science standpoint. There is studies showing that donepezil can attenuate amyloid deposition and mitochondrial dysfunction in the brains of mouse models for Alzheimer's. And there is also data showing that it can activate AMPK in the brain. We are wondering if it potentiates contractions. And actually a colleague of mine down the floor who studies neuromuscular physiology had published a paper in 1997 where they were looking at muscle contraction in rats. And they had a cholinesterase inhibitor on board. And when they did, the force production for a given stimuli was much higher than the vehicle or a different neurotransmitter that was in the mix. So this led us to think that maybe somehow donepezil potentiates contractions, and maybe that somehow aids in preventing the skeletal muscle mitochondrial dysfunction that occurs with Alzheimer's. So our next steps with this project is one, we're currently doing RNA sequencing of the skeletal muscle to see if we can get any clear targets for why Alzheimer's would lead to deficits in mitochondrial function. And then also examine how donepezil may be having a positive effect. And we also have some pilot funding to explore this in a preclinical basic science model of advanced aging, the CISD2 knockout mouse, where we're going to explore if donepezil exercise or donepezil plus exercise can prevent advanced mitochondrial dysfunction in muscle. Okay, so the next line of research I'm going to talk about involves exercise and pharmacotherapy. And most people have heard about statins. These are lipid-lowering drugs that are really important for people with high cholesterol. They have a great deal of benefit for people that have diabetes, high cholesterol, or have had a cardiovascular event. And a lot of individuals, as they're aging, are told by their doctor, hey, take this pill and you need to start exercising. Oops. Okay, I'm trying to advance my slides here. There we go. So statins are, of course, a class of lipid-lowering medications. In 1999, it was about 12.5 million people took these medications. By 2003, it was double that. It's now estimated that more than 40 million users worldwide. And in 2013, the American College of Cardiology and the American Heart Association came out with recommendations that would even greatly increase the amount of statin use. And what was important about this, these recommendations was that it was pushed that they should be used more as a primary preventive tool, not so much once you had type 2 diabetes or if you had a cardiovascular event, but as a primary prevention for younger people. And so how do statins work? Well, they block HMG-CoA reductase, the rate-limiting enzyme for making cholesterol. But what they also do by blocking cholesterol is they block mevalonate, which is important for ubiquinone or coenzyme Q10, which is a electron transport chain component. It's an electron carrier in the electron transport chain. And what a lot of people don't realize is that when statins inhibit cholesterol, they do inhibit cholesterol synthesis, but because of the inhibition of synthesis, the liver cells actually become basically hungry to restore cholesterol levels. And so there's a lot of reuptake of cholesterol in circulation. And that's 50% of how they lower cholesterol. And so there's been a lot of data over the last 15 years showing that statins are not harmless agents. They actually have been linked to insulin resistance and to an increased risk for type 2 diabetes. The risk is small, but it is significant. And honestly, the risk is about the same for the positive effects of preventing outcomes in people who don't have any other risk factors. So this is an issue in the field, and a lot of people are really hesitant now to take statins because they've heard about this. And so, of course, statins do have positive outcomes. They greatly have reduced cardiovascular death rates in at-risk patients. Probably the best population that they've had the biggest effect is people with familiar hypercholesteremia who basically can now survive into adulthood normally because of statins. And there's been other positive pleiotropic effects. There's some evidence that they're anti-inflammatory in people with congestive heart failure. But then they have a number of negative effects that have just been, over the last 10 years, more closely monitored in the literature. Some people experience pain, exhaustion, fatigability. There's evidence of some muscle damage and myopathies and injuries. There's evidence of mitochondrial abnormalities. And then I already talked about insulin resistance and risk for type 2 diabetes. And in rare cases, very rare cases, there's risk for rhabdomyolysis. So when I was junior faculty at the University of Missouri, we started an exercise trial. This is back when the metabolic syndrome was a hot thing to study, and it was kind of argued how important the syndrome was. And what we wanted to do was do a simple exercise intervention comparing statin therapy to statin therapy plus exercise to exercise alone. And really this was, I'll have to admit, an internal grant. I was wanting to get exercise interventions, muscle biopsies, different things up and going. That was the whole reason behind this. And we were really testing to see if there was one advantage over another for these three different kinds of interventions. And I'm really only going to show data from the statin therapy plus exercise versus exercise. I'm not going to report the statin therapy only. This is the subject characteristics of the people we tested. There was 18 in the exercise only group and there was 19 in the statin plus exercise group. This was not a blinded study. This is before I knew how to do those kinds of studies. So everyone knew what they were on. And they had to have, we were trying to target people who probably should be on a statin but weren't on one yet. And the primary findings were, and we saw this early on, the primary findings were that VO2 peak, or the improvement in aerobic capacity induced by exercise, was blocked if individuals were taking statins. So this is the absolute data. Here is the delta response to the 12-week exercise intervention. I want to highlight that this was a 12-week exercise intervention at our exercise fitness center. So this was all monitored by undergraduates and graduate students. And here's the time to exhaustion. So both VO2 peak and time to exhaustion were reduced if they were on statins. We took muscle biopsies and we measured markers of mitochondrial content. We weren't doing respiration at the time, but we measured markers of mitochondrial content. You can see the exercise group had the expected increase. This is the delta response. We measured a number of other mitochondrial content markers, and you can see the same general trend across most of these. So there's been a bunch of questions in the literature. Do statins cause discomfort and cause people to be less active because of that? And do they advance the loss of aerobic capacity? And so we began to think about this. If individuals have a low aerobic capacity as they get older, does something like a statin drive that down further and advance this even further? Another question is, if someone is high fit and has high mitochondrial function, are they less, or do they have more protection against statin-induced changes in VO2 max? Things that are completely unknown. And I want to highlight that there is really good data that fitness and statins can coexist. This is a study from Kokonos published in Lancet in 2013. This is collected in the VA population in California, and where they've done a whole bunch of aerobic capacity testing over the years. And this is data that can be interpreted kind of however you want. They have 20,000 men over 15 years. They looked at mortality over a 10-year follow-up. And what they showed were that statins and fitness both reduce mortality independently. They showed that the high fit people on a statin had the greatest protection. And they also showed that moderate fitness was independently protective for mortality. So here you can see the lowest fit group versus the highest fit group, the entire cohort. This is the same data with just the patients taking statins. Here's the data with patients not taking statins. But where you really see interesting results is in the hazard ratios, where you can see the high fit statins individuals had the lowest hazard ratio. But you see incremental improvements whether you're on a statin or what level of fitness you have. So this is interesting data. And it suggests, the reason I show this is I'm not saying that high fit people can't be on a statin and maintain high aerobic capacity, because there's evidence that it doesn't always affect that. Now, other groups had started to emerge with data showing that people on statins had lower mitochondrial function. Steen Larsen in the Copenhagen group, led by Fleming Della, had showed in a cross-sectional study that complex 2 or complex 1 and complex 2 respiration was reduced in people that were on statins, and it was about 50% lower respiration, about 50% of max. And then we had started talking with Dr. Daryl Neufer's lab at ECU, and Terrence Ryan was a postdoc in Neufer's lab at the time. And they had been doing some studies on statins in primary cell culture models. And so they decided to conduct a study, a pilot study, dosing young people with 80 milligrams a day of Atorva, which is a dose that is commonly used in the United States. And then they studied those individuals at baseline, 2 weeks, 4 weeks, and then 6 weeks. And they looked, they took biopsies at each point. And what I want to highlight here is that you can see by day 28, about 1 month out, that there was a dramatic decrease in skeletal muscle respiration. Calcium retention also tended to go down, and insulin sensitivity went down in approximately 6 out of 8 subjects. They also used NIRS to measure mitochondrial function and saw a deficit there as well. And so in the field, there's quite a few papers now looking at how statins may cause mitochondrial function or dysfunction. it's pretty well thought that there's evidence of it being a complex 2 inhibitor. There's some evidence of it being a complex 3 inhibitor. A lot of the theories in the field relate to statins' ability to block mevalonate synthesis and thus lead to reduced coenzyme Q and ubiquinone levels. And anecdotally, I've had several people tell me that are on statins, if they take coenzyme Q, they feel better. And there is some animal data to suggest it may protect against mitochondrial dysfunction. However, this is really good data to suggest it's not all coenzyme Q mediated, because this is permeabilized muscle fibers that are just treated for 10 minutes with Atorvastatin or simvastatin. And you can see in that 10-minute period that there's a reduced respiration. So it cannot be all due to a reduction in coenzyme Q. So we have an ongoing trial. Unfortunately, this is a randomized control double-blinded trial, so I cannot show you any data today, but I wanted to talk about what we're trying to do. We're 3 years into the trial. We have 2 years left to go. Aim 1 is to determine the impact of low and high dose statin therapy on skeletal muscle mitochondrial function, insulin sensitivity, and fitness. And we're doing a dose time duration randomized control. And what we want to do is test a high dose versus a low dose versus a placebo. And we're doing testing at baseline, 1 month, 6 month, and 12 months. And we want to see, what we hope to see is that there's less of a deficit with a 20 milligram dose. And so that can be safely tolerated. And then that would tell the field that that's a good dose to use for prevention. Aim 2 is to, again, redo the exercise trial, the 12-week exercise trial. And again, we hope that exercise adaptations are normalized with the lower dose. And again, we think that this would influence the field. So this is a 2-site trial. Dr. Neufer and his lab at East Carolina are primarily doing Aim 2, and our lab here at KU Med in Kansas City is primarily doing Aim 1. And I just want to show, talk a little bit about the difficulties in doing a clinical trial where you're also trying to do basically hardcore basic science. So we are doing screening, then we do baseline testing in 3 to 4 visits, and we collect all these different outcome measures. The muscle biopsies we're doing, and then we're doing about 4 to 5 hours of respiration studies on mitochondrial biology. We do biopsies again at 1 month, 6 months, and 12 months. We have glucose tolerance tests along here. We have VO2 max tests along here. We work with a cardiovascular lab led by Sandy Billinger, and that lab does the NERS for us, and they also do pain assessment and physical function testing. So this is a really big study that takes a lot of people working on it, and I'm hopeful that we get some really interesting data within a couple of years. Sorry I can't share more today. So this is Aim 2. And I kind of want to move on to just a real quick, another commonly used drug for metabolic health, metformin. And of course, if you get type 2 diabetes, you are told to, or the American Diabetes Association guidelines are that you should get on a metformin and you should get on a statin. So the statins is to control lipids and reduce cardiovascular risk. And the metformin is for prevention and treatment of type 2 diabetes, and it basically lowers glucose. Interestingly, both show evidence of being mitochondrial electron transport chain inhibitors. I showed you data earlier on statins, but there's really good data that metformin also inhibits mitochondrial respiration, and it seems to mostly be mediated by being a complex 1 activity inhibitor. And in fact, that might be how it lowers glucose. It may disassociate or uncouple energy production in the liver, thus lowering gluconeogenesis in the liver. And there's a lot of papers out there on how metformin works, and it's a very contentious field. It's really not known, even though it's been used as a drug for glycemia for over 40 years. So what about metformin and exercise? So 2 big studies have come out in the last year or 2 showing negative effects of metformin with exercise. One study I'm going to show a little data from is from Dr. Ben Miller's group when he was mostly at Colorado State. Now he's at Oklahoma Medical Research Foundation. And then another paper came out from Charlotte Peterson's group showing that metformin blunted muscle hypertrophy in older adults using resistance exercise to offset atrophy. I'm only going to show data from the aerobic exercise study here. And basically what Ben did, or Dr. Miller did, was show the exact same thing we saw with statins, that individuals participating in a exercise intervention, if they were on the placebo, they had a normalized improvement in VO2 max, but if they were on metformin, the response was abrogated. They also took muscle biopsies and measured respiration studies in the mitochondria of the permeabilized fibers. And you can see that metformin reduced all the improvements in exercise induced respiration. I'm not going to show it, but they also showed that metformin blunted the ability of exercise to improve insulin sensitivity. So this is a really interesting thing in the field, that we have a lot of people who are sedentary and at risk for metabolic disease and cardiovascular risk are put on these drugs, and they're also told to exercise. And we have data that metformin and statins both have negative effects on skeletal muscle mitochondria, both block exercise adaptations, and there's a lot of need to do more studies on dosing, timing, and susceptibility. And then we hope that randomized controlled trials can inform treatment decisions. Dr. Miller got NIH funding, and he is now doing a bigger randomized controlled trial studying the effects of statins plus exercise. We recently wrote a review in the physiology journal from the APS, and Dr. Miller is an aging expert, and what we talked about in the paper was the effects on health span. And the whole idea is that you want to prolong health span as long as you can before you have to go into a disease period. You want to compress this disease period as much as you can. And the question is whether pharmaceutical treatments prolong the health span, or if they block the ability of exercise to prolong that health span. Okay, getting closer to the end here. The last thing I want to talk about are a few slides on our work on fatty liver. Unless you've been living under a rock, you've probably heard about fatty liver. It's the new big metabolic condition affecting millions of Americans and people across the world struggling with obesity. Steatosis is the first step, and that is the excess storage of fat in the liver when it's greater than 5% of the liver comprised of fat. And that has all kinds of risks associated with it. But then, of course, once you have steatosis, you're at much greater risk for NASH, which is steatosis with inflammation and fibrosis. And then once you have NASH with fibrosis, you have much greater risk for cirrhosis. So about 20 to 30% of people who get steatosis will progress to NASH, and then another about 10 to 20% of those individuals will go to cirrhosis. Even if you just have steatosis, though, you have hepatic insulin resistance and greater risk for type 2 diabetes. You have greater lipids. You have risk for carcinoma, and then, of course, liver failure. So it's a bad condition. And this is an old slide from a review from 2004, but it still rings true. We know that fatty liver is somewhat largely a condition of positive energy balance and obesity. And so we know the individuals that are more obese are more at risk. People with bad diets, high in sucrose and fructose and fats, are at more risk. And there's really good data to show that both activity and fitness status impact risk. And importantly, there's really good data that even if you're obese, that if you're active and you have high fitness, your liver's protected from fat storage. At the end of the day, hepatic fat storage, excess hepatic fat storage, is due to these four things. You either have excess delivery of fat to the liver that the liver can't handle. You have excess synthesis of fat through de novo lipogenesis and triglyceride synthesis. You have reduced ability to export triglycerides out of the liver, or you have suppressed fat oxidation. And what I want to really highlight here is this is where we can really start to see the impact of skeletal muscle metabolism on the liver. We know in individuals that have skeletal muscle insulin resistance that they're going to have higher insulin and glucose levels in circulation. And those factors then drive transcriptional programming in the liver that promotes more fat storage and less fat oxidation. Recently, Scott Rector and I wrote a review for diabetes on all the different ways in which we think exercise protects the liver from excess fat storage. And you can see all these pathways here. The green indicates known effects. The red indicates limited evidence or hypothesis. And we did it in both a fed and a fasted condition because there's different things going on in the two different conditions which we think impact liver fat storage. But what I want to emphasize in both conditions is that if you have high functioning skeletal muscle, that's highly insulin sensitive and can highly oxidize fat, that you'll see one of the ideas of how exercise protects against fatty liver is that you have a lot of the substrates being channeled to muscle, so it saves the liver from having to deal with them. And that's probably a really oversimplification of how it occurs, but it probably does play a pronounced role. So for example, in a fed condition, the adipose lipolysis would go down, but the dietary fat and carbohydrates coming in would primarily go to the muscle to be oxidized or to be stored, rather than all being delivered to the liver, where the liver would have to deal with it. I'm gonna get back to this link here in a second in a more direct way. So back to the HCR-LCR rats. We've been using this rat model of high and low aerobic capacity for about, I guess, 12 years now. And what we've shown with multiple studies is that the HCR rats, just like humans, the HCR rats with high aerobic capacity are protected against steatosis. So this is a study where we did a 3-day high-fat diet. We call it the Thanksgiving at Grandma's house. And in this kind of model, you can induce steatosis in just 3 days with a high-fat, high-sucrose diet. You can see the LCRs already have higher steatosis, and they get greater steatosis and liver triglyceride accumulation. Here is the histology where the HCRs are protected. So for the first few years of funding for this project, we really tried to phenotype how are the HCRs protected from steatosis. And one of the things we really thought a lot about is mitochondrial energetics in various tissues. And either our lab or other labs has clearly shown that the HCR rat have higher mitochondrial function in adipose, in liver, and in muscle. And the reason why we have higher mitochondrial function in liver is to support endurance exercise. I'm going to get to that in a second. But here's some of the salient findings we've had in the HCR versus the LCR, and we think these are all important. Number one, the HCR have higher activity. They also have higher resting and non-resting energy expenditure. So even when they're not moving, their energy expenditure is higher, and they kind of live in an uncoupled state. They also have a better control of energy intake. They oxidize dietary lipids better. They have more insulin sensitivity, and they have completely different ways of trafficking substrates to different metabolic tissues. And then in the liver, we found big differences as well. So it's not just a systemic phenotype. There's liver changes as well. I'm not going to talk about those as much today, but they have higher mitochondrial fat oxidation and respiration. They have greater transcriptional responses to changes in dietary substrates, different epigenetic profiles, and they even have different mitochondrial post-translational modification profiles. Now, we did clamp studies in the HCR-LCR rats on animals on a control diet and animals on a high-fat diet. And what I really want to point out is that the HCRs are able to maintain their insulin sensitivity. The LCRs already have lower insulin sensitivity, and it's driven down by just the 3-day high-fat diet. And what you can appreciate here is that when we look at glucose disposal in both skeletal muscle and in adipose tissue, the HCR have increased glucose disposal. I don't want to spend a bunch of time talking about this response with the high-fat diet. That is something we've never seen in any other animal model where glucose uptake actually increases on a high-fat diet. But we also looked at glucose trafficking to glycogen, and it showed the same effect. So this is real. But my point is that the animals have higher insulin sensitivity, and they have a greater capacity to traffic glucose into muscle. And I think that's important, and I'll get to that in a second why. And then also, if we feed them a high-fat diet and we trace the fatty acids with radiolabeled palmitate and oleate, which allows you to look at fatty acid partitioning into various tissues, as shown here, you can see that there's no real big difference in liver trafficking of fatty acids. But what you'll see is that the LCR preferentially traffic dietary fatty acids to adipose, where the HCR preferentially traffic fatty acids to muscle. And we think this is a big benefit because once it goes into muscle, it's not coming back out. It's going to be oxidized and used in the muscle. Again, preventing the liver from being exposed to it. Okay, so why would aerobic capacity increase mitochondrial content and function in muscle? We think it has to do with gluconeogenesis. It's well known that when animals or humans exercise, gluconeogenesis goes up over time, glycogenolysis goes up, and total hepatic glucose production goes up. And the way this works is that our system is built intricately in a way that the ATP turnover in our metabolic tissues, including the brain and the muscle and the liver, are tightly controlled or linked with how much glucose the liver makes. And as soon as ATP demand goes up and muscular work starts, the liver can sense this mostly through changes in glucagon and insulin, and quickly release glucose or increase gluconeogenesis to match this and keep us euglycemic in our glucose levels level. And the way that the liver fuels this process is by oxidizing fatty acids to make ATP, and those fatty acids come from lipolysis in adipose tissue. So if we think about this in an intact system, and this is showing it in the HCR versus the LCR, that the HCR has greater hepatic fat oxidation paired with greater hepatic gluconeogenesis, paired with greater ATP turnover and glucose utilization in skeletal muscle. And so the way we think about this is that the muscle is pulling on this whole system, pulling on gluconeogenesis, which the gluconeogenesis is relying on fat oxidation and lipolysis of fatty acids. And this all makes sense because animals that can run longer have greater hepatic glucose output, and we've shown repeatedly the HCRs have greater PGC1 alpha, which controls both mitochondrial fat oxidation and gluconeogenesis. All right, so I'm wrapping this up here pretty soon. What I want to just highlight on this slide is that the role of mitochondrial function in fatty liver is actually quite controversial. And I won't go through all this data. I'm just going to say the following. There's a lot of evidence in humans that when humans develop steatosis, they don't have reduced mitochondrial function. Instead, they have compensatory increases in TCA cycle flux, in oxygen consumption, and in respiration. Because it's being overwhelmed with fat, the liver is trying to upregulate fat oxidation and mitochondrial function to compensate. And so this is what was shown by Sean Burgess in a really nice animal study where they use isopotamer analysis. So they combine with NMR, and by doing that, they can really measure trafficking of substrates through all these hepatic biochemical pathways, and they actually can measure flux of nutrients. And so what they did was an 8-week, a 16-week, and a 32-week high-fat diet. The black bars are the high-fat diet. And they used this isopotamer analysis and metabolic flux measurements to look at TCA cycle activity in the liver. And you can see that by 16 weeks, it goes way up in the high-fat diet fed animals. They can measure gluconeogenesis. You can see gluconeogenesis goes up, and they can measure anaplerosis, and you can see anaplerosis is going up. And this is primarily through the increased activation of pyruvate carboxylase, which is turning pyruvate into oxaloacetate, which then drives into the TCA cycle and basically is like throwing gas on a fire. And so with a high fat steatotic condition, you have the high fat diet and the obesity pushing on the liver, pushing these substrates to the liver. And the way the liver handles this enhanced anaplerosis and oxidative metabolism is to make more glucose and gluconeogenesis, which then increases glucose in circulation. And they've also had really good data that this then leads to oxidative stress and inflammation and probably plays a major role in liver disease. So we went back and we worked with Dr. Burgess at UT Southwestern and his postdoc Stan Deja, and we asked, can we do these tracer techniques in the HCR-LCR rats? And they were nice enough to collaborate with us. And here's kind of the experimental paradigm. We use U13C propionate and 3,4,13C glucose. And they did these fluxes for us. And you can see that the HCR has greater flux through almost every metabolic pathway in the liver. And they can take this data and they can actually calculate TCA cycle flux and gluconeogenic flux. And you can see it's higher in the HCR than the LCR. Now, I want to highlight, now we've got a situation where the disease state, steatosis, is showing the same phenotype as the metabolically protected state, the HCR, or the high aerobic capacity. But what we think is critical is this data I've already showed you earlier, that there is higher gluconeogenic flux, but it's paired with greater glucose disposal in muscle. And so again, the muscle is pulling on this, or at the very least, the muscle has glucose disposal rates and ATP turnover rates that are on par with what's occurring in the liver. So there's a pairing between these two metabolic organs. And so the way, I really need to get a graphics person to help me show this better, but the way we think about this is that an exercise or high aerobic capacity situation, there's a pull and there's a pairing between the metabolic functions of the liver and the muscle. But that in a sedentary, low aerobic condition on a high fat diet, you have a pushing of nutrients into the liver. There's compensatory increases in fat metabolism and gluconeogenesis in the liver that are not paired with elevated glucose uptake in muscle. And that leads to then hyperglycemia. And we've done have some unpublished data that even just running in Sprague-Dawley rats increases T-cell cycle flux. This was collected 24 hours after a last running bout, and we're continuing to follow up on these studies. So in conclusion, exercise, healthy condition versus steatotic disease condition, both induce elevated hepatic mitochondrial respiration, T-cell cycle flux, and gluconeogenesis. But the healthy phenotype seems to be linked to a pull from skeletal muscle, or at least a better pairing with skeletal muscle metabolism, including insulin sensitivity, greater ATP turnover capacity, or overall energy expenditure rate at the whole body level may be playing a role as well. And I want to just highlight that the last two lines of research I've been showing are actually somewhat related. There's pretty good data that impaired mitochondrial function in the liver, or at least excess fat storage in the liver, may compromise the liver's ability to catabolize drugs like statins. So in that scenario, if you have low aerobic capacity, you have impaired mitochondrial function in your liver, or you have a steatotic liver, when you take a drug like a statin, which is catabolized in the liver, theoretically, the muscle, if it's not degraded properly, the muscle is then exposed to higher levels of statin, which could cause greater toxicity. And there's actually really nice data in animal models to suggest this occurs. All right, with that, I'll wrap it up. I want to acknowledge all the people who have worked on these projects over the years or are currently working on these projects. This is my lab from actually a couple of years ago. They're a great working group. These are my collaborators here at KU Med, including the people in the lab. Previous collaborators at University of Missouri. Sean Burgess at UT Southwestern has helped me a lot with these tracer approaches, which is really advancing what we're doing in the lab. And I have some other collaborators, including at Michigan, where they provide the HCR, LCR rats, and then Daryl Neufer at ECU with the statin study. And our funding agencies. So with that, I'll take any questions.
Unmute. Thank you very much, John. What a great job. Lots of interesting questions. I didn't stop writing the whole time. So what we'll do is we'll take some questions, please, through the Q&A forum, if you don't mind, rather than the chat. And my job is to just really ask those questions. And let's start with the first one. Okay, so Chris Perry, regarding the donepezil study. Is the drug metal-based? If so,
yeah,
it might be attracted to heme of the ETC, which could mean it's a direct mitochondrial inhibitor. What do you think about that?
Well, so we actually showed that donepezil improved mitochondrial function, or at least in this cross-sectional analysis of individuals, it was associated with improved mitochondrial function. So I don't think it's a mitochondrial inhibitor, but I can look into that and find out. I don't know if it's metal-based.
Yeah. Okay, fair enough. You know, I spend, while I'm waiting for a few more questions through Q&A here, I'll just ask a couple more, and I'm sure Chris has a few more he can ask too. You know, I'm looking for drugs that enhance the exercise response. You know, like that to me is, and it's interesting from your perspective, you're looking at the drugs that inhibit the exercise response,
Yeah.
like, you know, the metformin and of course, you know, hugely popular drugs, statins and metformin. So I got to commend you on this. This is work that's really very important. And on the other hand, I'm sort of looking at the opposite kind, not with that much success, to be honest, yet, but looking for it, you know, like, so something that enhances, something that synergizes with exercise. I just throw that out as a comment.
Yeah, I agree. I mean, and I'd really love to. I really haven't had a chance to talk about the donepezil data with very many muscle physiologists, just to think if this is just a harebrained idea. I remember I talked to Marcus Bamman about it, and he thought that that drug would probably change receptor affinity for acetylcholine. I think he mentioned it may upregulate acetylcholine receptor expression, but we don't know. But this seems to me like potentially a drug that could potentiate every motor unit activation by allowing the acetylcholine to stay around longer. Obviously, you got to have it. I did a little reading, and it sounds like it inhibits about 50% of the acetylcholine diesterase. So you still have diesterase activity, obviously, because otherwise people would lock up.
Yeah. Okay, so Mike Riddell asks, comments, the issues of statins are a real pickle. Sure, it's possible that the peak aerobic capacity may be negatively impacted by statins, but it looks like you might live longer to exercise. How can you find a way to tweak the system to make the drug better for those of us who like to exercise?
So I think what I would do, and I don't know how I would ever get this kind of project funded, but I would, if I had patients and all the money and time in the world, I would say, okay, first I want you to go through a cardiac rehab program, 8 to 12 weeks. We're going to really boost your aerobic capacity up. Then we're going to put you on a statin. Because I think, there's probably no problem adding the statin once you're already an exerciser. And, and so if you start here and a drug knocks you down, that's bad. But if you start up here and a drug knocks you down a little bit, it's less bad. And then also, we've been thinking lately that, the hepatic ability to catabolize the drug may be increased by exercise. And I have a student or a research assistant professor in the lab who's going after that. So if exercise can actively enhance the ability of the liver to catabolize the statin, and then the muscle's exposed to less, the muscle really shouldn't be exposed to any, then that also could be a potentially good thing.
Mike has added his comment that his VO2 dropped from 85 down to 70. VO2 max went with 1 year on statins. He believes that it can affect your VO2 max. Okay.
He's not the only highly trained person I've heard that from.
Tim Moore is asking, I'm wondering if, given the heterogeneous nature of the cells in the liver, are these phenotypes from hepatocytes or another cell within the liver? Maybe immune cells. I guess there's a lot of heterogeneity in the liver cells. So,
yeah, the liver's a mess. So the liver has hepatocytes, has endothelial cells, sinusoidal cells, has Kupffer cells, has stellate cells. And we typically isolate the mitochondria and measure respiration. We think we have primarily hepatocyte mitochondria, but we probably have contamination from other pools. And so that's a great point. And there are now labs doing single cell RNA-seq. So you actually label multiple hepatocytes and label multiple stellate cells and look at the RNA signatures of those. And I think those are studies that need to be done. And then you also have zonation in the liver. There are areas of the liver, it's very much analogous to muscle fiber type. You have areas of the liver that are more, have more blood flow, more oxygenated, have more mitochondria, and then other areas that have less oxygen, less blood flow. And those cells behave differently as well.
Right. I got a feeling from your description of the NIH clinical trial that you're really anxious to talk about it, but you can't talk about it. You must be excited about the outcome of that one.
We are. It's a struggle to recruit. It's a struggle to recruit for that study. We're trying, number one, to find people who probably should be on a statin, but aren't yet. So that makes it hard. And then there's just a lot of visits. And a lot of invasive procedures, to be honest. So it's a hard trial to do. And I doubt we meet the recruiting goals that we're trying to do for the power analysis we did, but I hope we can get close. And of course, we were really ramping up last fall, and then COVID hit. And so we lost about 5 or 6 months there. So I'm hoping the NIH gives us another 6 months to a year of funding to go longer.
Yeah, I mean, that should happen. I hope that happens for you. I was just interested in that Alzheimer's disease mouse model. Can you describe that a little bit? I hadn't heard of that before.
Yeah, it's called the CISD2 knockout. It's a you ever heard of Mito? I think it's some kind of I wish I could describe it for you better, David. It's a model that's down the hall. It's one of about 15 or 20 different models that show really advanced aging due to a mitochondrial phenotype. I can send you a couple papers on it, but especially the females are a little bit better to study. The males around 8 to 10 weeks of age just really deteriorate, and by 20 weeks of age basically look like a really old animal that's has low muscle strength, low muscle tone. So it's a good animal to study something like this in a short timeline.
Yeah, right. Sounds good to me. Anyone else with any other questions for Dr. Thyfault this afternoon?
I see Michael now says 50 to 45 instead of 84 to 70.
Yeah, I was giving him the gears there. He's not taken lightly to it, I think. Here, here's an anonymous question. If anyone has looked at some of the preclinical statin alternatives and their effects in combination with exercise. For example, bempedoic acid, which is a liver-specific inhibitor of ACLY. Can you speculate on what the effects might be?
So I have not looked at that, but an area of research that we're pursuing, which I did not share here at all, and there's been one other paper in the literature that exercise in rodents actually increases fecal loss of cholesterol and bile acids, which both come out, through ACLY. And so we have this hypothesis that one of the ways exercise might protect the liver from excess fat storage is actually loss of energy through fecal matter, and that might be a way that exercise lowers cholesterol too. And so we've looked at that, but we have not looked at it like an ACLY inhibitor.
Okay, very good. Anyone else out there who'd like to ask a question? Let's see. Mike Gridell has asked, I like the push-pull model for liver muscle metabolism. How can you try to prove that?
Yeah, I've spent a lot of time trying to think about that. Like, is there a hexokinase inhibitor that will just target skeletal muscle, or can we somehow knock down GLUT4 just in skeletal muscle, in an exercise condition and induce that phenotype? And I've talked to people like Dave Wasserman and different people, and we can't think of a good way to do that. Now, Wasserman has approached this a little bit with his hexokinase heterogeneous mice, or heterozygote mice. So they have he's done both full knockout and 50% knockout in muscle of hexokinase, which then of course affects glucose uptake in muscle pretty dramatically. And that data pretty much supports the same idea, but he's never looked at the liver phenotype to see if it changes the liver phenotype.
Mm-hmm. Yeah.
So it may just be a good analogy to give lectures and something I never really can prove.
Oh no, sounds like a good hypothesis to test, actually.
Yeah.
Yeah. Okay, folks, I think we've asked Dr. Thyfault plenty of questions. John, just stay with me for a minute. And
Okay.
Not seeing any more questions, I guess we'll put an end to the meeting. I want to thank everybody for coming and attending. And of course, I want to thank John for his great presentation and for being with us here today. So that's the end of the meeting, folks, and have a good afternoon.
Thanks for having me.